Okay, welcome to today's lecture. I hope you're all doing well. My name is Owen Donoghue, and I hope you enjoyed the previous lecture on an introduction to the clinical significance of nucleic acid therapeutics, with a focus on how they could be used against inherited diseases. Today, we're on to our second lecture in this theme, where we look at the clinical significance of nucleic acid based therapy in the context of cancer. So, unlike the conditions we focused on last week, that are due to inherited or germline mutations, cancer is not generally thought of as an inherited condition. It occurs as a consequence of mutations that occur in certain cells during our lifetime, referred to as somatic mutations. But it's important to remember that you can have genes that put you at a higher risk of developing cancer. And a good example that you may have heard of would be mutations in the BRCA1 gene that increases the risk of breast cancer. But even in cases like this, the occurrence of cancer to a large extent is still dependent on additional mutations occurring during our lifetime, rather than being directly due to mutations that we're born with. But like inherited diseases, cancer can be treated with nucleic acid based therapy, and this is what we're going to focus on today. As part of this, we'll mainly focus on mRNA based cancer vaccines, which are an important emerging treatment modality against cancer. And I hope that from this you can gain some kind of an insight into the in vivo action of this kind of nucleic acid based therapeutic intervention. As the name suggests, cancer mRNA vaccines essentially represent an immunotherapy based approach to tackling cancer. And as some of you may be aware, in recent years, immunotherapy based approaches have really had a big impact on cancer management. And this makes sense, really, because one of the things that the immune system is good at, of course, is finding things that don't belong in our bodies and eliminating them. So, just like in the case of viruses or bacterial infections that are detected and eliminated, our body does have the ability to do the same thing with dysfunctional or damaged cells, such as cancerous cells. So, to understand how we can use mRNA as a therapeutic molecule to activate the immune response against cancer, it's good to understand first how, under normal circumstances, the immune response protects us against cancer. Then we talk about what goes wrong with this and why the immune response arrives at a point where it can't effectively eradicate tumors. And then finally, we learn about how understanding this has allowed us to use mRNA to reactivate the immune system against tumor cells. So, first of all, let's take a look at how the immune response protects us from cancerous cells all of the time. And there's a bit of immunology in this, but this is not an immunology course. And I'll talk you through what is a very, simplified description of this process that will provide enough context to appreciate the potential of mRNA based cancer vaccines. So, cells become dysfunctional all of the time in the body. And this is principally, but not always, due to mutations that occur during cell division. But as a result of these genetic changes, and sometimes epigenetic changes, cells may lose the ability to make correctly functioning versions of important proteins, or they may lose the ability to regulate important cellular processes through non coding genes, including processes like cell division, which needs to be very controlled for obvious reasons. But some cells that do lose the ability to control cell division any longer just keep dividing and dividing in a completely unregulated way. And these cells are basically cancer cells. And as this division continues, you get a large aggregation of cells, which we refer to as a tumor. And this uncontrolled division is usually avoided by cells engaging in programmed self destruction if there's something wrong, which occurs through processes such as apoptosis, for example, or alternatively, the immune system can eliminate these cells as well. So normally, this is not a problem. Between apoptosis and other forms of programmed cell death and the immune response, these cells are eliminated before they become a problem. And our immune system typically shouldn't attack our own cells. This is only something that happens if something is wrong with cells. Just as the way patrolling immune cells, such as dendritic cells, recognize infected cells, for example, through various non-specific and general signs of infection before any adaptive immune response. They can also recognize unhealthy cells, again, just through other very general characteristics that unhealthy cells exhibit, leading to phagocytosis, which is basically just the engulfment of cells. So, like any unhealthy cell, cancer cells can exhibit signs of stress and damage. Immune cells recognize these injured, stressed, or dying cells as they display what are referred to as eat me signals on the outside. And this is what leads to phagocytosis. And they can also engulf cell debris from dead cancer cells that may have died due to stress, maybe rather than programmed cell death. And just like when these patrolling immune cells, such as dendritic cells, engulf viruses or infected cells, after they engulf cancer cells, dendritic cells become activated or begin to mature, and they degrade these proteins and display bits of these proteins on their cell surface on MHC1 or MHC2 molecules, alongside co-stimulatory molecules, which they also begin to produce after this stimulation. In the context of infections, this normally occurs in response to pathogen associated molecular markers or cytokines that are released from surrounding infected cells. But in cases like this, where there is no infection, but where you just have damaged cells or cell debris, dendritic cells do this in response to stimulation from damage associated molecular patterns and or stimulatory cytokines that are either within or released by these unhealthy cells or present within the cell debris. So regardless of whether they are derived from an infected cell or a non-infected unhealthy cell, the purpose of this antigen display on the cell surface after phagocytosis is to allow them to be presented to T cells in the lymphatic tissue. So these antigens can be inspected to see if there's anything non-self present. And it's a useful default mode of action of dendritic cells or any antigen presenting cells, because when they encounter something in the presence of damage associated patterns, while it doesn't mean there is an infection taking place, it's possible that sometimes some of this damage could actually be due to an infection. So responding to cellular damage in this way, which is similar to the way that they should respond to an infection, is a very useful precaution. So any antigens that are taken up here are presented to T cells to screen for any non-self antigens, for example, from pathogens such as viruses. And when I say non-self, I mean anything that the body has not encountered before, so it has no tolerance against it, and the immune system won't accept the presence of anything like this in the body. And this is particularly useful in the context of cancer cells because, as mentioned earlier, cancer cells can arise due to mutations in proteins that lead to incorrectly functioning versions of normal cellular proteins. And if these mutated proteins look sufficiently different from normal proteins, they may be recognized as being sort of like something that is non-self. And these are referred to as neoantigens. And remember, with neoantigens, as these arise due to mutations that emerge during our lifetimes, we're not born with them. So our immune system should naturally recognize them as being new or non-self. So these neoantigens are distinct from other types of antigens that we are probably more used to hearing about that come from pathogens like viruses. But in any case, it's useful if the immune system can recognize neoantigens, because if they are seen as not belonging in the body, it will result in the body mounting a targeted and specific response against cells that express these neoantigens in order to eradicate them. In other words, the recognition of neoantigens as non-self initiates an immune response against cancer cells, just as it would against virally infected cells. So the main component of this adaptive immune response against cancer, or at least the ones that have received the most attention, are T cells. And specifically, I'm talking about subpopulations of T cells that are, by chance, capable of binding to these neoantigens via their T cell receptors. So after the recognition of these antigens via the T cell receptors, these T cells become activated, which is helped by the engagement of these co-stimulatory molecules, and then this is enhanced further by the release of cytokines, and this leads to clonal expansion of these specific subpopulations of T cells. And broadly speaking, there are two kinds of T cells that become activated through this process. Firstly, CD8 killer T cells that recognize neoantigen on MHC1, and CD4 helper T cells that recognize neoantigen displayed on MHC2. However, T cell activation, killer T cell activation is highly reliant on help from activated CD4 helper T cells. And through this process, CD8 killer T cells become licensed to kill, and they leave the lymphatic tissue to find and destroy any other cells that display the same or similar neoantigens on their surface. And they can do this because all cells, or inside all cells, all the time, all cellular proteins are constantly being degraded, and bits of them are being displayed on the surface on MHC1 for this purpose. So the repertoire of proteins produced by cells are always visible to patrolling CD8 killer cells. So in the context of cancer, these cells can find cancerous growths at various stages of progression by recognizing these neoantigens that are displayed on MHC1 molecules. And once they're recognized, they initiate the killing process. But all of this killing doesn't go unnoticed. It attracts dendritic cells that are stimulated again by the damage associated patterns, and in response to this, then they engage in phagocytosis. And just like outlined earlier, this is followed by antigen processing and presentation, leading to a point where they can again present the same or possibly new neoantigens or other tumor associated antigens to the adaptive immune system again in the lymphatic tissue, starting the cycle once more. So, through this, you can really begin to see just how useful the immune response is in terms of dealing with cancerous cells, especially if they're really good neoantigens that can be properly exploited by the immune system. Also, it's important to remember that in addition to this cell mediated response, in theory, it's also possible to have an antibody response against these kinds of mutated proteins. But this is reliant on specific circumstances, such as the mutated protein being secreted or being available on cell surfaces. And it also has to look different in its native undegraded form in order to be recognized by B cells as being non-self. And if so, these are degraded by B cells, and the resulting neoantigens are displayed on MHC class II alongside co-stimulatory molecules and are presented to activated CD4 helper T cells. And if these same antigens are also recognized by these T cells, then you get an optimum antibody response against the mutated protein. Although antibodies can, under the correct circumstances, be indirectly useful against cancer cells, as I implied earlier, this is only really useful against mutated proteins that are secreted or if they're on the cell surface. But the exact contribution of antibodies towards eradicating cancer cells is not really that well studied. It's believed that CD8 cells, in contrast, which engage in the direct killing of cancer cells, are generally more effective at this process. But it's good to be aware that there can be a B cell antibody response too, which may be useful under some circumstances. But overall, regardless of the mixture of adaptive immune responses that occur in response to cancer, this leads to the formation of long-lasting memory T cells or B cells even against these neoantigens that can respond immediately if the same neoantigens are encountered again, without the need for this elaborate activation process involving the prior presentation of antigens to T cells in the lymphatic tissue, as these cells can be already present in the peripheral tissue and already licensed to kill. And it's something that's really useful at preventing the formation or recurrence of tumors. So this is a very, very simplified account of how the immune response against cancer works. And our current understanding of this is, of course, a lot more nuanced than what I've described here, but it gives you an idea of how useful the immune response can be against cancer. And indeed, it's how we believe most cancerous cells or small tumors are eradicated at the very early stages. And it's really a good way to get this done. So if it fails, leading to the development of cancer, turning this response back on can be very, very beneficial from a therapeutic perspective. So this leads us to the question, why might this fail? Why exactly this happens is not fully clear, but at some point in early tumor formation, cells that make up the tumor are just not recognized by the immune system as being a problem that needs to be taken care of. Among other things, it's possible that neoantigens may not be sufficiently different from normal self antigens to elicit a strong adaptive immune response to prevent tumor formation. Also, there might be no neoantigens at all. As mentioned earlier, the uncontrolled cell growth, it could be something that happens due to dysregulation of normal cellular processes due to maybe epigenetic changes, for example, or maybe even changes in non-protein coding genes. As a tumor grows and there's an enormous amount of cell division taking place, maybe also combined with a lot of suboptimal DNA repair processes, these cells may accumulate more mutations, leading to the eventual emergence of neoantigens, which is of course useful. But by then, a new problem typically develops. Often in order to sustain itself, or as a result of poor blood supply, or as a combination of both, a low oxygen or low pH environment often forms around tumors, which really inhibits the normal function of immune cells. And this is referred to as the tumor microenvironment. The exact nature of different tumor microenvironments vary from tumor to tumor, but one common characteristic is this inhibition of immune cells. In addition, tumor cells may produce immune response inhibitors themselves, or they may attract immune suppressing cells. So even if neoantigens do emerge as a tumor progresses, the immune system may not be able to exploit them. So while it's not always clear why the immune response fails initially to respond to cancer cells, why it fails once a tumor actually forms is something that's much more clear. For example, in solid tumors, there's certainly no shortage of damage associated patterns or damage associated molecular patterns for dendritic cells. Although most cells are dividing rapidly, many of them are under severe metabolic stress. With a lot of cells dividing, of course, you also get a decent amount of cells also dying or in a state of necrosis, especially at the center of solid tumors. So there's plenty of material here for dendritic cells to engulf and to be stimulated by. But in this environment, the inhibition of proper dendritic cells function means that they may not engage in phagocytosis, or if they do, they may not end up expressing important co-stimulatory molecules on their surface. So if they do end up engulfing cells and somehow managing to still migrate to the lymphatic tissue, they're going to be poor activators of T cells once they arrive there. So this is one of the main ways that the tumor microenvironment may inhibit processes that are important for starting a normal adaptive immune response against cancer cells. However, inhibition of this response can also occur at the later stages of this process too. For example, if CD8 killer T cells are somehow activated by dendritic cells and they arrive in the tumor microenvironment, they themselves can become inactivated in this environment for many reasons. For example, one process that has received a lot of attention is the inhibition of CD8 killer T cells by high levels of immune checkpoint proteins, for example, PD-L1 on cancer cells. So when T cells encounter cancer cells and they're stimulated by these cells via their PD-1 receptor, it leads to what's referred to as T cell exhaustion, which causes them to be essentially ineffective. So when CD8 killer T cells enter the tumor microenvironment, you don't get this expected killing of cancer cells that are displaying the correct neoantigens that they recognize. So this is what can go wrong, or it's a very quick summary of what can go wrong. But how can mRNA-based intervention help? Well, the idea behind this is very simple in principle. Instead of relying on dendritic cells obtaining neoantigens within the tumor microenvironment, which as we know can be a place where they're severely inhibited, the mRNA can be used to deliver neoantigen to dendritic cells while they're outside the tumor microenvironment. So out here, in the absence of any inhibition, they can properly mature. So the mutant protein is expressed in these cells once the RNA enters. Some of it is degraded, and the resulting antigens are displayed on the cell surface on MHC molecules. And regarding the display of neoantigens on MHC1 molecules in dendritic cells that do receive mRNA, this is something that's going to occur as a natural consequence of the translation of this mRNA in these cells, but also as a consequence of the inherent immunostimulatory nature of the mRNA itself, even when you use modified bases, or the inherent immunostimulatory nature of the delivery system, or through the action of an adjuvant, if present. This immune stimulation results in the expression of essential co-stimulatory molecules that are required then to properly engage with T cells. And it also triggers MHC2, or the display of antigens on MHC2 molecules on the cell surface by activating autophagy related processes in these cells. So as a result, then you get this proper dendritic cell maturation without the need for damage associated molecular patterns. And this is important as the display of antigens on MHC2 largely requires phagocytosis first. For example, phagocytosis of dying cells or cell debris in order to obtain undegraded antigen first. But this alternative route towards MHC2 display in these dendritic cells is really useful in the context of mRNA-based vaccination, where there will not be any undegraded soluble form of the antigen available, as it's just mRNA that's being delivered. And however, it's important to remember that soluble undegraded versions of the neoantigen may eventually become available for phagocytosis if mRNA transfected cells undergo apoptosis as a result of immune stimulation, which is something that can certainly happen. So either of these two things could lead to this essential MHC2 antigen display on the surface of these dendritic cells. So it's essentially no different here from what happens during mRNA vaccination against COVID-19. So then, after display of all antigens, in other words, neoantigens and non-neoantigens, they'll be presented to T cells in the lymphatic system. And of course, only neoantigens will be recognized by T cells, as there should be tolerance towards the non-neoantigens. So after neoantigen recognition and activation of T cells, as described earlier, you get an expansion of tumor-killing CD8 killer T cells and increased infiltration of these cells into the tumor microenvironment. And of course, this environment may still be somewhat inhibitory towards CD8 killer T cells, for example, from immune checkpoint ligands described earlier, leading to T cell exhaustion. But the idea here is that maybe the increased T cell infiltration overall may offset this in some way. But this inhibition from PD-L1 could also be offset by combining this type of mRNA therapy with checkpoint inhibitor treatment. And this is something which generally takes the form of antibodies that can bind to and block this PD-L1 on the surface of cancer cells, reducing the occurrence of CD8 killer T cell exhaustion in the tumor microenvironment, which of course could lead to increased killing of cancer cells. And in theory, either with or without these checkpoint inhibitors, this mRNA-driven CD8 killer T cell response should help eventually destroy the tumor, despite the fact that dendritic cell inhibition is still ongoing in the tumor microenvironment, which should, under normal circumstances, prevent this beneficial response from happening in the first place. So you go from a situation of having tumors where there is no useful immune response occurring, which are referred to as cold tumors, to a situation where you have tumors where these processes have been kickstarted once again, and these are referred to as hot tumors. So in cancer immunotherapy terminology, this is often referred to as turning cold tumors hot, and it's the main goal of this kind of an approach to cancer therapy. Even in the early stages of this response, this mRNA-driven immune response should act to reduce the tumor's ability to sustain the tumor microenvironment, or it should at least make it more difficult for it to spread, with small metastases with the same neoantigens being much more vulnerable to these same killer T cells that will recognize them, as they don't have their own well-developed tumor microenvironment yet. This process also leads to the subsequent formation of tissue-resident memory T cells that can also react to future new metastases, eliminating them immediately in the peripheral tissue, without the need for dendritic cell activation and migration from the lymph nodes first. That's how cancer mRNA vaccines work, and hopefully it gives you a better insight into their action in vivo and their sheer potential. And one thing you might be wondering is, how are neoantigens actually selected? First of all, it's important to remember that each patient may have different neoantigens or combinations of 10 to 20 or more neoantigens. So it makes sense to establish what neoantigens exist in each tumor first and then synthesize mRNA to deliver these mutated proteins with these neoantigens to dendritic cells, as we described earlier. And what we're talking about here, in essence, is really the first real progress towards what is described as personalized medicine. And in terms of establishing what neoantigens exist in cancer cells that are not present in normal tissue, it's possible to take samples and compare tumor tissue to normal tissue by comparing transcriptomes, for example. In other words, by identifying almost every mRNA that's present and by extension what protein it produces and what ones differ from normal tissue due to mutations. And this can be combined with mass spec of protein content also. And these are things that are becoming cheaper and cheaper to do, and it's something we get done here on a routine basis in our lab when we're looking at virally infected cells versus non-infected cells. So by comparing transcript and protein profiles from tumor and normal tissue, you can identify what protein mutations are unique to tumors. And you get to a point where you know what mRNA you need to deliver to dendritic cells to kickstart an anti-cancer adaptive immune response, as outlined earlier. And in practice, this process of antigen identification is a lot more complicated than this, of course. What I have here is a highly simplified account of this process, but it will allow you to understand the principle behind the approach for sure. And also, it's important to point out that it's not totally necessary to rely on stimulating an immune response against neoantigens. As mentioned earlier, not all cancers have associated neoantigens. Some may be caused by overexpression of non-mutated proteins. These are called tumor-associated antigens. And unlike neoantigens, of course, they're also present on healthy cells, but they're just massively overexpressed in tumor cells. And these can also be identified via the same processes that I mentioned there on the previous slides. Because they're clearly normal-looking proteins, that are also going to be present on healthy cells, T cells that can recognize tumor associated antigens and bind to them strongly are usually eliminated during T cell development due to what's referred to as central tolerance. Either that or they're eliminated later due to what's called peripheral tolerance. So these processes are useful as it eliminates T cells that can attack healthy cells. However, T cells that bind weakly to these self-antigens may survive these processes, for example, avoiding central tolerance or peripheral tolerance, and some of them can end up in the lymph nodes, and they will bind, albeit weakly, to these self-antigens when they're presented to them on dendritic cells. And normally it's thought that there should be no big response by these T cells unless you get a lot of these antigens being delivered to the lymph nodes by dendritic cells that are also expressing co-stimulatory molecules. So this leads to some sort of expansion of these T cell populations against these tumor-associated markers. And it's assumed that this is what does happen in the case of cancer vaccines that are based on tumor-associated antigens rather than neoantigens. As the T cell binding that we're talking about here is weaker, these cells have a much weaker interaction with normal cells, of course, but their impact on cancer cells is still greater due to a greater number of binding sites, leading to increased contact opportunities and more sustained contact time and increased stimulation to engage in killing. So it's speculated that this is why these T cells that target tumor associated antigens have a bigger impact on tumor cells compared to healthy cells that also have the same antigens. So the ideal tumor-associated antigens to target for therapeutic use will be present at really, really low levels on normal cells, but at extremely high levels on cancer cells. And the greater the difference between the two, the better. And there are many known tumor-associated antigens that are overexpressed on cancer cells. As mentioned earlier, tumors from patients can be sampled and analyzed to identify the overexpression of known tumor associated antigens in order to determine what tumor associated antigen mRNA vaccines may be best to use. So, in terms of antigens for cancer mRNA vaccines, tumor associated antigens represent a much more off the shelf approach, relying on pre made mRNA vaccines against a broad range of known tumor associated antigens that can be applicable to a broad range of patients and represents a good alternative to neoantigen vaccines. But from this introduction to both neoantigens and tumor associated antigens, as you can probably understand, there are advantages and disadvantages to either approach, and they're summarized here in this table. I'm not going to go through it in any great detail, you can certainly read it yourselves. But briefly, the main advantage of stimulating an immune response against neoantigens is that it's likely to lead to greater efficacy, as T cells that bind very strongly to these antigens are not going to be subject to central tolerance. And because the same antigens won't be present on non cancerous cells, there should be a less unintended impact on other tissues. While the opposite is, of course, true for when stimulating a response against tumor associated antigens. However, on the other hand, the more off the shelf nature of mRNAs against tumor associated antigens means that they can be delivered to patients quicker and they're much more cost effective as there's less customization involved in their manufacture. And there are plenty of trials underway with both approaches. For example, BioNTech are conducting trials with both types of cancer vaccine, whereas Moderna's cancer vaccine pipeline appears to be totally focused on neoantigens. Intuitively, the neoantigen approach should work better, but the patient customization aspect makes it more technically difficult to expand out to large amounts of patients at the moment, and it comes with a much greater price tag, of course, because there's so much customization involved. But in the long run, one thing is inevitable, and that is that the cost of identifying patient-specific neoantigens through transcriptomic and proteomic profiling will come down. So we can see this happening every year in our lab when we want to do similar things with virally infected tissue or cells. So while this neoantigen approach is not so straightforward at the moment, it should become more and more streamlined and more cost effective to do over time. So it'll be certainly interesting to keep an eye on this. And also, it's important to take note that some of these trials are also testing the impact of the co-administration of these immune checkpoint inhibitors that I outlined earlier. And this may help counteract CD8 killer T cell exhaustion in the tumor microenvironment. In several cases, they're testing co-administration of immune checkpoint inhibitors versus monotherapy with mRNA vaccines or immune checkpoint inhibitors alone. But it's important to remember that immune checkpoint inhibitor therapy is only useful if the immune checkpoint blocking does not cause issues elsewhere in the body, where fully functional immune checkpoint molecules are still important, of course. And in these cases, these are being delivered as pre-manufactured antibodies, as outlined earlier. But if you remember from last week's lecture, it should be possible just to deliver mRNA for these antibodies and let the body's own cells produce and secrete them. And indeed, such approaches are under development at the moment, but they're at the preclinical stage currently. And I think this potential use of mRNA cancer vaccines and mRNA-based immune checkpoint inhibitors in combination is a good example of how different kinds of mRNA therapy in cancer can be used in very complementary and synergistic ways. And it's a great testament to the sheer versatility of mRNA in terms of its therapeutic potential. So this brings us to the end of this week's talk, a little shorter than last week, and I hope it gives you a broader understanding of the therapeutic potential of nucleic acid-based therapies beyond the inherited medical conditions that we discussed last week, and where this area could potentially be heading in terms of how RNA-based therapy is a major factor that facilitates the progression towards a new era of truly personalized medicine. So that's all from me for this week. I hope this has given you more of an insight into how the immune system protects us from cancer, why it fails, and how it can be reactivated again using mRNA cancer vaccines. So I'll be back next week with another talk where we'll focus on new and exciting emerging technologies in this area. Until then, take care.